Hi, my name is Shruthi, and I'll be talking about the role of aberrant splicing in Parkinson's genes and emerging splicing therapies. I'm currently a senior in Mountain House High School in California, and my mentor for this project is Matthew J. So I started my research on aberrant splicing in Parkinson's disease after witnessing my grandfather's battle with PD. It was devastating, and I was determined to understand why this was happening. By researching the role of alternative and aberrant splicing in Parkinson's, I hope to contribute to ongoing research and share potential splicing therapies to ultimately improve the lives of patients. So, what is Parkinson's? Well, it's a progressive neurodegenerative disorder characterized by the loss of dopamine producing neurons in the brain. There are many motor and non motor symptoms associated with this disease, with the most common being resting tremors, observed in 80% of the cases, and bradykinesia. These symptoms seen on the screen collectively emphasize the complex degeneration defining PD progression, significantly impacting daily function and quality of life. While there is currently no cure for PD, various treatment approaches can significantly improve a patient's quality of life by managing the motor and non motor symptoms associated with this disease. The primary treatment for Parkinson's involves levodopa to replenish dopamine levels, which is often combined with carbidopa to reduce side effects like dyskinesias. Dopamine agonists and enzyme inhibitors are also used to delay the need for levodopa and slow dopamine breakdown, but their use may be limited by side effects, especially in older adults. There's also deep brain stimulation, which is a surgical and reversible alternative. DBS involves the implantation of electrodes in specific brain regions to deliver electrical impulses. This helps control movement related symptoms such as tremors and rigidity. Physical, occupational, and speech therapies also help with the disease, and they aid in improving gait, voice disorders, and cognitive functions. It is important to note, however, Parkinson's disease treatments currently focus only on managing symptoms and improving quality of life rather than curing the underlying condition. So, research is ongoing into potential disease modifying therapies aimed at halting or slowing down the disease progression today to ultimately introduce a cure for Parkinson's disease. So, now I'll be talking about aberrantly spliced genes that contribute to PD. So, first there's, oh, sorry, I think I skipped one. So aberrant splicing is when alternative splicing goes wrong, and it's one cause of Parkinson's. So the brain is particularly reliant on alternative splicing, which is a process to create a variety of protein isoforms. And it's the brain's complex neuronal networks demand a vast array of proteins. However, this also makes the brain vulnerable to splicing errors or aberrant splicing. So, aberrant splicing can result in various consequences, including protein misfolding, aggregation, and impaired cellular function, contributing to the development of neurodegenerative diseases, like Parkinson's disease. Um, Parkinson's disease is a prime example with several disease-associated genes, including PARK2, LRRK2, SNCA, and PINK1, which all exhibit aberrantly splicing patterns. So now I'll be talking about the aberrantly spliced genes that contribute to PD. So PINK1 is a vital protein that helps remove damaged mitochondria through a process called mitophagy. So PINK1 achieves this by phosphorylating and stabilizing parkin, a ubiquitin ligase that tags dysfunctional mitochondria for degradation. When PINK1 proteins become truncated or non-functional due to aberrant splicing, it cannot properly regulate mitophagy. This leads to an accumulation of damaged mitochondria, causing increased oxidative stress and inflammation. Consequently, this contributes to the progression of PD. Another gene, which is PARK2 or Parkin, is an E3 ubiquitin ligase that directs specific substrate proteins for degradation by tagging them with ubiquitin. Its structure, characterized by distinct domains, is crucial for enzymatic activity. This process is vital for maintaining cellular homeostasis and mitochondrial function. So, aberrant splicing of PARK2, particularly in exons 2 to 4, generates generates dysfunctional isoforms lacking in essential domains, stopping its ability to attack proteins for disposal. These defective Parkin proteins accumulate within the neurons, leading to cellular stress and damage, particularly in the substantia nigra. This disruption of protein degradation and mitochondrial function contributes to the development of PD. So, in the image shown on the slides, it kind of shows how Park2 and Pink1 work together. And basically, as they go through aberrant splicing, it makes a lot of mistakes in how the cell is actually supposed to function, and that can lead to PD. So next, I'll be talking about SNCA and LRRK2. So the SNCA gene produces alpha-synuclein, which is a protein important for dopamine homeostasis and other cellular functions. So this protein has a special structure that helps it work correctly. So in normal cellular processes, asynuclein interacts with phospholipids and dopamine transporters, and it inhibits phospholipase D2. And this maintains neuronal function and synaptic plasticity, which is really important for the brain. So when the SNCA gene is aberrantly spliced, it can make an abnormal version of alpha-synuclein. So this abnormal protein sticks together and accumulates in the neurons, particularly in the substantia nigra, leading to cellular toxicity and neurodegeneration. So these clumps can damage brain cells, especially those involved in movement, and this damage leads to progressive neuronal loss characteristic of PD. LRRK2 is another protein essential for various signaling pathways. So LRRK2 plays a role in protein phosphorylation and protein-to-protein interactions, autophagy, mitochondrial function, and synaptic vesicle trafficking. So it has like a lot of roles in the brain, so this is one of the most important genes. So mutations in LRRK2 can disrupt these functions, often altering its kinase or GTPase activity. So this disruption can lead to impaired autophagy, resulting in the accumulation of damaged cell components. And these cell dysfunctions can disrupt the normal neuronal signaling and lead to Parkinson's disease. So now I'll be talking about the possible therapies for PD addressing aberrantly spliced genes. So first is CRISPR-Cas9, which is a precise gene editing tool. So it functions as a molecular system capable of altering specific DNA sequences. So, and this process involves two key components, which is the guide RNA, also known as the gRNA, and the Cas9 enzyme. So this gRNA identifies the target DNA sequence where changes are needed. It directs the Cas9 enzyme to the correct location, and then the Cas9 enzyme cuts the DNA at the specified location. The cell's natural repair systems then mend the DNA break. There are several potential applications of CRISPR-Cas9 in Parkinson's treatment, like gene correction, gene silencing, and gene delivery. Although it's important to note, while CRISPR-Cas9 holds immense promises, it's still an emerging technology, and significant challenges such as delivering the system safely and effectively to the brain and ensuring precise edits without unintended consequences need to be overcome. Next, there's RNAi. So RNAi works by first identifying the target gene, then small interfering RNA molecules are designed to complement the gene's messenger RNA. Next, the siRNA molecules are transported into the brain cells, and the siRNA molecules bind to the target mRNA, initiating its degradation and preventing protein production. So this therapeutic effect aims to reduce abnormal protein levels associated with PD and slows disease progression. So inhibiting the production of harmful proteins, RNAi has the potential to slow the degeneration of brain cells, alleviate Parkinson's symptoms, improve patients' quality of life. While RNA also holds promise, challenges such as effective siRNA delivery to the brain and potential off-target effects require further investigation before clinical application. And these are my works cited. Thank you.
Thank you so much. Another very complex topic explained so well. I appreciate you sharing your work. Let's see, I am curious of the potential gene editing therapies that you shared, which one do you think is the most near term to actually making it to clinical?
Um, I would say RNAi just because it's reversible, so there's like a higher chance for it to be on market like sooner. But I feel like there is more research going into CRISPR as of now. But yeah, it's really in the early stages. There aren't many like case studies going on with them, but it's the forefront of healthcare and it's really important.
Absolutely. And thank you so much for enlightening us